All right, so what we're going to do is review signal transduction and hopefully go over some of the pieces that may be giving you some trouble. Um, the first thing that you have to look at when we are going to talk about signal transduction at all is whether or not your ligand even needs a signal transduction mechanism. That is to say that when the ligand itself is lipophilic, there is no reason that you would need a signal transduction mechanism because those ligands pass right through the cell membrane. They're either going to have receptors in the cytoplasm or actually within the nucleus, where those ligands can actually direct transcription. To do all that takes a little bit more time, so you see a higher lag time. We're talking about ligands here like steroids that can do things like that. Actually tell the cell, this is the gene that you need to transcribe right now. On the other hand, if the ligand is lipophobic, if it cannot cross the cell membrane, you're going to have a signal transduction pathway because it can't cross the cell and yet it's charged with giving the cell a message. And so the only way to do that is it's going to have to bind to some kind of receptor and it's going to cause some cellular response. So we're going to look at those next. The different options that we see, the different ways that we're going to look at signal transduction throughout the semester include these kinds of things. What that first messenger, that ligand shown in yellow, what that can actually do is it can bind to receptor channels directly, as is shown on to the far left. So when the ligand binds to the channel, it can either cause the channel to open or close. So that channel there could be, say, for ions, could be some kind of exchanger. It's going to be really important when we get to the nervous system that those could be channels for ions having to do with sodium, which will cause the cell to depolarize. Next, second to the left, you see a receptor enzyme. So the ligand can actually bind to a receptor that is directly attached to an enzyme on the intracellular side. So as long as the ligand is bound to the receptor, that enzyme is either functional or is not functional. So by changing the shape of the enzyme, that ligand can actually control whether the enzyme is catalyzing reactions or not. We also see the ligand here binding to a G protein coupled receptor. We're going to see that in more detail here in just a second. As you can see, that's probably one of the more involved signal transduction pathways that we're going to look at. And then to the far right, we have an integrin receptor, and this is how a ligand can actually change the shape of cytoskeletal elements. So the first one that we showed in class was the tyrosine kinase signal transduction system. And here you see the ligand at the top binding to a receptor. Again, its job is to carry a message to the interior of the cell when it itself cannot cross the cell membrane. So it binds to this receptor, and the receptor, the whole complex there, changes shape, and that tyrosine kinase becomes active. So you see the active binding site there as long as the ligand is bound to the receptor. And what the tyrosine kinase does is it pulls phosphate off of ATP and phosphorylates a protein. So that is an example of substrate level phosphorylation, right? And a kinase is any enzyme that does that. This one in particular, because of the way that its structure is, we call it a tyrosine kinase, but all kinases phosphorylate like this. Okay, and lastly, we have the ligand turning on a G protein system. And I know this looks kind of scary, but if we just sort of take you through the steps here, it's really not so bad. And we're going to see this over and over again, so you're going to get used to it. So the first thing that happens is the ligand, again, cannot cross the cell membrane, so it binds to that receptor, as you see in number 1. That thing looks kind of like a snake. It passes in and out of the membrane itself, so it's anchored in there. When the ligand binds, it causes that G protein that you see to change shape. And an alpha subunit, which is the one to the right, will dissociate. It actually comes off of the G protein and it just floats through the membrane. And where it binds, where it has a binding site, is on this enzyme, the adenylyl cyclase. Adenylyl cyclase has a weird name, but it's an enzyme, right? And it's attached to the membrane. And when the alpha subunit of the G protein binds to it, it changes shape. Of course. And what the adenylyl cyclase does, it acts as an amplifier enzyme. So as long as the alpha subunit is present, what adenylyl cyclase does is it takes not one, but two phosphates off of ATP, leaving you with cyclic AMP. And I showed you the structure of that in class. That's adenosine monophosphate. That little C there just has to do with the fact that the phosphates and the way the oxygens are takes on a sort of a little ring kind of shape. So they call it cyclic AMP. Cyclic AMP is the second messenger, okay? And so as long as that alpha subunit remains bound to adenylyl cyclase is going to grab ATPs and just churn out cyclic AMPs. It is 1-to-1, okay? I don't like the way that she drew that, but one ATP, you strip off the terminal two phosphates, you get one cyclic AMP. What cyclic AMP does is it activates protein kinase A, as you see here, and kinase A, just like all kinases, it phosphorylates things. And here we get into the variability of what kind of cell this actually is. So all these proteins that are already made get phosphorylated. That's the way of the cell activating them, of doing something. And so then you get a huge, very, very loud cellular response. And you notice that it is amplified. So this is an example of both a cascade, right? Because you see one step causing something else, and you also see the amplification. So you see how powerful that one little ligand, that first messenger is. You only need one of them to cause a huge, fast cellular response. So this video is a really good example of signal transduction and how epinephrine causes liver cells to release glucose.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of cAMP from ATP. The cAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
Pretty cool, huh?